The order that nucleotides have in DNA is what it's called a sequence, and the techniques and methods used to know that sequence are called sequencing methods. Knowing the order of nucleotides is a tool with countless applications. At the beginning of sequencing, obtaining the sequences of a fragment of 5 or 10 nucleotides was a very expensive and complicated process. However, scientific and technological advances have allowed many DNA sequences to be had today.
The chemical method was proposed by Maxam and Gilbert in February 1977, 10 months before Sanger published the enzyme sequencing method. They developed a technique for sequencing DNA molecules. The method determines the nucleotide sequence of the DNA that is radiolabeled at one end and cuts it with a chemical reagent at each base. Using polyacrylamide gels and separating these fragments by size, the sequence of the molecule can be determined. This method has the ability to sequence 100 bases of DNA. These are the steps for this method. In the image you can see the number 1, that is obtain the single-stranded DNA. The number 2 is to add a 32p to 5 end. Uh, Number 3 is to cleave and add specific nucleotides. The number 5, the number 4 is differently sized DNA strands. The number 5 is electrophoresis through high-resolution acrylamide gels. And finally, number 6 is to deduce the DNA sequence. You can apply this method in many different ways. One of these is the HLA typing.
This is used to match patients and donors for bone marrow and cord blood transplants. These are markers found on most cells in your body. HLA matching promotes the growth and development of new healthy blood cells and reduces the risk of a post-transplant complication called graft versus host.
Another one is the DNA footprinting. This method is used to identify specific DNA binding protein sites. It can not only find the target protein that binds to a specific DNA, also identify which sequence the target protein binds to. The protein binds to the DNA fragment, protecting the DNA's it I DNase one cutoff binding site. After cutting, fragments of DNA molecules remain that there is no radiolabeled band corresponding to protein binding site on autoradiography of polyacrylamide electrophoresis gel. The fingerprint measurement method has high specificity, accurate positioning, and is widely used.
1977, Sanger's enzymatic sequencing emerged. This enzyme technique involves making many copies of a region of DNA, only that it is based on the controlled interruption of in vitro DNA replication. Certain reagents are required to perform this sequencing. DNA polymerase enzyme, a primer, the four nucleotides of DNA, the DNA mold that will be sequenced, and the special ingredient, the deoxy or chain terminators for the four nucleotides each marker in order to be recognized later. Unlike the deoxynucleotides have no hydroxyl group in carbon 3 of the sugar ring, and once a deoxynucleotide is added to the change, there is no longer a hydroxyl on which more nucleotides can be added. So, to carry out the Sanger sequencing methods, all ingredients must be combined into one tube, but the deoxynucleotides are put in much smaller quantities than normal nucleotides, and once all are together, the mixture is heated to denature the DNA mold and then cooled so that the primer can join the simple chain mold. Then the temperature rises to that the DNA polymerase can synthesize new DNA from the primer And DNA polymerase will add nucleotides to the change until it randomly adds a deoxynucleotide instead of a normal one, so that the change ends and will be marked particularly depending on the base it carries. And when a certain number of cycles end, the tube will contain fragments of different lengths, and is particularly guaranteed that the deoxynucleotides has been incorporated into each of the DNA sample positions. And when the reaction ends, the fragments are separated through gel capillarity electrophoresis, pacing by its sides. The smaller will pass more quickly, so each fragment crosses a line, and a laser will illuminate it, allowing detection of the associated marker. In this way, the sequence of the original DNA fragment can be reconstructed nucleotide by nucleotide from the markers recorded one after the other one by the detector. So, two main applications of each of these technologies. The first one, enzymatic methods, was in the Human Genome Project. Sanger sequencing was used to determine the sequences of many relatively small fragments of human DNA. The fragments are aligned based on the overlapping portions to assemble the sequence of the larger regions of DNA. And the other application is, although the genomes are currently sequenced with other methods that are faster or less expensive, Sanger sequencing is still widely used to sequence individual pieces of DNA, such as fragments used in the DNA cloning or those generated through PCR.
In order to provide an example of a sequencing machine, we present the PacBio sequencing tool. This sequencing machine is powered by single molecule real-time or SMRT sequencing, and it can take up any sample type, ranging from viruses to vertebrates. SMRT sequencing works by ligating adapters to the once extracted and isolated double-stranded DNA sample. This is to make a circular template so the polymerase can work continuously. At the core of the sequencing machine, we have the SMART cell, which contains millions of tiny wells called zero-mode waveguides, or SMWs. This is where the singular DNA molecules will be isolated and start a replication process with the help of a DNA polymerase. As the polymerase works on the replication process, a different wavelength will be transmitted to distinct each nucleotide that has been added, and that signal is registered in the machine to uncover the DNA sequence. This process has an accuracy of 99% due to the millions and millions DNA molecules that are being analyzed at the same time.